Alright, so now today we are going to look into the DNA replication. Now, did you know that the DNA replication is like a ultimate copy machine for life, churning out perfect genetic blueprints? So, because of this, we are diving into this high-yield USMLE topic to help you crush those biochemistry and genetics questions. So, let's get it started with the bite-sized high-yield facts. Alright, now let's talk about DNA replication basics. A super high yield for step 1. First off, replication always happens in the 5' to 3' direction for both DNA strands. Think of it like reading a book from left to right. The leading strand is synthesized continuously, nice and smooth like a highway drive. But the lagging strand, it's a bit choppy, made in short bursts called Okazaki fragments. This process is semi-conservative, meaning each new DNA molecule has one original strand and the one new fresh one, kind of like keeping an old favorite shirt paired with a new one. Now, here's the deal. Replication is more complex in eukaryotes than the prokaryotes because of the larger genome. But the enzymes, they are pretty similar across the board. Now for step 1, this is gold. Expect questions tying DNA replication to biochemistry pathway, so know it cold. Alright, now let's move to the next slide for the juicy details. Alright, now let's zoom into where the magic starts, the origin of the replication and the replication fork. The origin of the replication is a specific DNA sequence where the whole process kicks off. In prokaryotes like bacteria, there's just one origin, simple and straightforward. But in eukaryotes like us, there are multiple origins to speed things up for our massive genome. These origins are often AT rich, like the Tata box, because A and T pairs are easier to zip than G and C pairs. Now think of it like opening a flimsy zipper. At the replication fork, the DNA splits into a Y-shaped zone where the action begins. Both the leading and the lagging strands are synthesized at the same time, working like a well-oiled machine. Why does this matter for step 1? This is a core genetic concept. You will see it pop up in questions about mutations or replication errors. Nail those details and you're one step closer to acing those exams. That's your high yield dose for DNA replication for USMLE Step 1. Keep these concepts locked in and you will be ready to tackle those test questions. If you have found this helpful, smash that like button and subscribe for more USMLE high yield bites, and I will see you next time.